My name is Calvin Carpenter, and today we're going to be discussing a serious problem that has yet to really be paid attention to in today's society. So, before we get started, I have to ask you a simple question. Do you like bats? I hope you do, but if not, maybe I can help you to reconsider. Bats, like bees, are keystone species, ensuring such things like insect control, which they end up saving farmers around North America $14 billion in pesticides, and they also help with pollination. So without them, we wouldn't have foods like bananas, chocolate, or tequila. And they also help with seed dispersal, which helps with reforestation. But there are a lot of threats to bats, such as loss of habitat, wind turbines, and white nose syndrome, a disease caused by a fungus called Pseudogymnoscus destructans. This disease started in Albany, New York in 2006 and spread its way across America and is yet to be stopped, and really nobody is paying attention to it because they're bats, who cares? But ultimately, some people identified different bacteria that actually inhibit the fungus. The problem with that was the study was done in Canada. And if we want to ultimately bring in something into America or into different regions, it needs to be local and just in case if we introduce something new, it doesn't cause a bigger problem. And so ultimately, this is a continuation study, and I collected environmental samples back last year in a region to where, in priority caves where people were researching, in order to actually identify the bacteria if there was those known inhibitors that were identified in the Canada study. And I did confirm that there were those bacteria that were identified that inhibit P. destructans, but also identified other bacteria that were unknown to inhibit P. destructans. They'd never been tested before. So again, this is where I collected those environmental samples in priority areas where several research organizations were coming together, and this was kind of the hot spot of where everybody was. And but ultimately, the research question is, obviously, there's no known cure for white nose syndrome for bats, but they have identified those bacteria. But the problem is they haven't identified how those bacteria actually inhibit the fungus, because we have to identify the tools or the mechanism the bacteria is using in order to get an effective mitigation plan. And so the stars of today's show are first, of course, Pseudogymnoscus destructans, who we're obviously going to try and inhibit the best way we can, and ultimately identify the tools of Curtibacterium bacillum, which was unknown to inhibit P. destructans, and ultimately we're going to identify if it even inhibits, and if so, how it does, and then also B. subtilis, which in the Canada study was the best known inhibitor out of all the environmental samples they collected. So, first hypothesis is, does Curtibacterium even inhibit P. destructans? And so that's the hypothesis, Curtibacterium does inhibit the growth. And for this study, all the tests that I ran were using potato dextrose agar, which is just a media that grows fungus very well, but it also grows bacteria. And then ultimately, all the tests, I'll explain which test I use and how I worked with it, but ultimately it was put in an incubator at 13 degrees Celsius and incubated for 14 days, and then that's when I calculated the area of growth or inhibition. So for this study, for this test specifically with Curtibacterium, I ultimately did what I call a tic-tac-toe, but it's called a cross-streak inhibition assay. And ultimately, it's the parallel lines here are the fungus, and then parallel lines going horizontal are Curtibacterium. And ultimately, you can see there's no inhibition. But this is still relevant because this tells us we have to be choosing which bacteria is going to be best and how we're actually going to use it effectively. But since there was no inhibition, there was no further analysis conducted or statistics. But again, that brings us to the second point. Does Bacillus subtilis actually inhibit? We do know it does, so how does it inhibit? For this test, since we already know that it does inhibit, we have to go a little bit more advanced to actually identify how it inhibits. So for this test, I ultimately streaked two parallel lines right next to each other. One side's the fungus, and the other side is the bacteria, Bacillus subtilis. And we put them at various lengths apart in order to identify what range the bacteria has. Because if it's really close, that tells us something. If it's really far away, that tells us something. So ultimately you can see that here. It's kind of hard to see, but this is where the Bacillus subtilis was streaked, and then this is where the fungus, and I used image J in order to analyze the measurements from each side of the line. Now, we're looking for symmetry. If there's symmetry, that means there's no inhibition coming from Bacillus subtilis, because that tells us that nothing is affecting this side of the fungus. But ultimately, we can see with the results that there was significant inhibition, with the blue lines inhibited, on the left side of the line, ultimately telling us that the bacteria is doing something to the fungus. But it only goes up until about 12 millimeters. And we can see that with the statistics here, saying that when alpha was set to 0.001, which is essentially just the confidence level, and the lower the number of the alpha, the higher the confidence level, which means it's good. And ultimately, at 12 millimeters, it was sort of statistically significant, only with alpha set to 0.01, not as high as the others. And then, of course, at 15 and 18, there was no statistical significance, showing that the bacteria has an inhibition at a range at around 12 millimeters. Now, this is crucial because this tells us, oh, something is happening, the bacteria is producing something that inhibits the fungus. So ultimately, that led us to the next hypothesis of Bacillus subtilis extracellular components inhibiting P destructants, stuff on the outside of the cell. And ultimately, this led us to the initial stages of protein purification. Ultimately, what I did was I suspended the cells in a liquid culture in order to effectively get those extracellular components, stuff on the outside of the cell. And I used turbidimetric determination, which essentially just tells me the essential growth and optimal range of bacteria to liquid media. And then from that, I can maximize how much extracellular components are within the solution. And then from there, I spun it down really hard in a centrifuge and then collected the bacterial pellet, which will be important later. But for this study, for this test, we're using the supernatant, which has that extracellular component, stuff on the outside of the cell. And then ultimately stamped that onto a fungus, just like I did with the tic-tac-toe. And ultimately we can see that I used image J to analyze the intersection of the fungus and the extracellular components, and we can see that there was significant inhibition with alpha set to 0.001. So obviously there's something going on the outside of the cell. Well, logically, if there's something going on the outside of the cell, it also has to be going on in the inside of the cell. So now we have to analyze the intracellular components of the bacteria. And so ultimately, I resuspended that bacterial pellet we saved earlier and resuspended in a PBS solution of phosphate buffered saline, which essentially just makes sure that the pH stays relatively neutral and doesn't hurt the bacteria or cause it to break apart. And then from there, I essentially washed the bacteria, got it cue ball clean to make sure there's no other extracellular components that are going to affect the later test. And then from there, I spun it down again, used electrophoresis in order to ensure that there was no extracellular components within it. It just detects if there is any. And then from there, spun it down again, then lysed the cells using a sonicator. Essentially, it uses sound to break apart the bacteria and then get that intracellular components within inside. And then did the exact same thing and stamped it. As you can see, did the exact same region of interest analysis as I did for the other one, but as you can see here, there was not much inhibition. And it did have significance, but only at 0.01. Still not as significant as the other one. But this tells us that, well, if something's on the outside, it has to be on the inside. But if it's not on the inside, then that tells us that the bacteria is producing something, not secreting something from the inside. So with further research, I ended up discovering that the bacteria produces volatile organic compounds. They're just gases that the bacteria produces that have antifungal properties. But again, these have not been tested against P destructants. So for this hypothesis, it's essentially the Bacillus subtilis volatile organic compounds inhibiting the growth of P destructants. So for this test, this was actually my favorite one, is that it was two Petri dishes that one had the fungus in a 5 by 5 millimeter plug, and then on the other side, it was streaked with Bacillus subtilis, and then they were inverted one on top of another. And you can see here, this diagram shows it, and ultimately we can see that there was significant inhibition from the volatile organic compounds, but it was only at 0.01, which wasn't as significant as others that I've shown. So ultimately, we had to do a little bit more digging. And what I discovered was, with more study, that in the presence of Fe3 ion, the bacteria actually produces more volatile organic compounds. So if we want to determine if the bacteria is going to produce volatile organic compounds and that's what's causing inhibition, logically we have to put that ferric ion within the media, and then if we see a growth in, or see a decrease in the growth of fungus, then that tells us logically that the VOCs are being produced more. So ultimately it was the exact same test that I ran, just with this time we actually just put ferric ion on the media with the bacteria. And you can see that here with the inverted plate, the only difference is that now the Fe3 ion is on the plate with Bacillus subtilis, and then that's set against the fungus. And ultimately we can see that there was even more inhibition when the Fe3 ion was introduced to the bacteria, showing, and then at a significance of 0.001. So this is obviously showing that the iron causes the bacteria to produce more VOCs, which causes significant inhibition towards the fungus. So that brings us to the conclusions. First, we can see that Cortobacterium didn't inhibit. It's unfortunate, but this tells us that we have to look in the right direction if we want an effective mitigation option. But it also told us that Bacillus subtilis actually inhibits the fungus at a range of 12 millimeters. Then, continuing on, we found that it was extracellular components that actually inhibit the fungus, but not intracellular. So with further analysis, we conducted that VOCs are the things that's causing the inhibition towards the fungus. And we can see that within the presence of the Fe3 ion, it produced even more VOCs, which causes even more inhibition. So for future research, we have to actually identify which VOCs are going to be the ones that are causing this inhibition, because VOCs is a very broad category. And so with that, we can actually identify what VOC is going to be best and then run a cytotoxicity test, which essentially is just going to make sure that it's not going to harm anything in the cave, any living organism in the cave. Because if we introduce something else that's going to be worse, that's a bigger problem we're going to have to deal with. And then using CRISPR, which essentially just genetically modifies the bacteria and ultimately has it just produce that VOC, we can ultimately use that in later caves and again, just produce an effective mitigation strategy. Thank you.
Thank you, Calvin. That's very informative. We have a few questions for you now that are about your research process, and you might have gone over some of this information already, but just do your best. The first question we have is, how did you determine which results generated by your research method were most important for informing your new understanding?
Well, first of all, obviously the Cortobacterium, I truly thought Cortobacterium was going to have some inhibition because it is of known antifungals, but the problem with that was there wasn't. But that told us again, like I said, that we have to really focus on which options are going to be effective. If Bacillus subtilis is the only one that's going to be effective, we really have to narrow down what the Bacillus subtilis is doing to the fungus. So I really found that just varying bacteria, if it inhibits or not, was very important.
How do your findings provide directions for future research in the field?
So as I said within the future research slide, we have to narrow down the category of volatile organic compounds because it is a very broad topic, and volatile organic compounds contains alcohols and sulfides, which if we were to introduce that in a cave, it could harm bats. So future research is going to have to find something that is effective towards the fungus, yet still safe for the environment of the cave.
How did your expert advisor facilitate your deeper understanding of the research process?
So with this, my expert advisor really informed me on what tests were going to be good. He kind of grounded what I was thinking because I really wanted to do a lot. And ultimately, especially with the Bacillus subtilis being streaked at various distances apart, I ultimately wanted to do all the distances, and I wanted to do all the bacteria that I identified. But to really get effective answers, we had to narrow down what we wanted out of this, and ultimately chose only 2 bacteria that seemed the most effective, and then ultimately chose the different various lengths apart that would still give us that information we wanted.
Terrific. Thank you very much.